This is Bernard Paulson. In this lecture, we will add a pathway to the coupled glycolytic and pentose pathway model discussed in lecture number 11. What we are adding to that simulator now is more than a pathway. It is going to be a series of reactions. And when we have integrated the two previous pathways together with what we are about to do in this lecture, we basically have formed a network, a metabolic network that forms the core of metabolism in the human red blood cell. So the outline of this lecture is as follows. We are going to look at the pathways that synthesize and degrade AMP. In the previous model, the input of AMP was fixed at a constant, was a forcing function, as forcing functions were introduced in chapter 6. In most pathways in human metabolism, there are inborn errors or genetic defects, and the pathway we are about to add, which is purine metabolism, comes with quite a few well-known inborn metabolic errors. As before, we will first look at the subnetwork that we are about to add. We'll formulate it, balance it, do QC, QA, simulate it by itself, and try to understand some of its characteristics. And what we do then is we integrate it with the two coupled pathways from the last lecture. The integration issues here are many, as there are many points of contact between the two modules being integrated. We will then do a dynamic simulation of 50% increase in the rate of use of ATP usage, which is the same simulation as we've been considering throughout the text. The lecture, like previous lectures, is a lecture on how to build simulators, how to grow them in scope, size, and content, and how to simulate them. What I will make some comments on at the end of this lecture is the fact that we have now built a network model that is a big portion of the key metabolic pathways in the human red blood cell. And just make some comments about where large-scale models can be used and what their characteristics are. So we're going to talk a little bit about some biochemistry up front in this lecture, as I tried to do in the previous ones, to help us develop some understanding of the background and enrich the material from a relevance standpoint. Here I have a screenshot from the book shown. This screenshot shows the purine nucleotide metabolism and its degradation, as well as for pyrimidines. We're going to be focusing on the purines, and we're going to be focusing on what's called a salvage pathway, which is the recovery of the nucleotides as they're degrading, like reaction 1 and 2 shown in this diagram. That odd molecule PRPP transfers a double phosphate bond to rebuild the nucleotides. As you see at the bottom of this chain of reaction, if the nucleotides fall apart into xanthine, uric acid, and so forth, they eventually end up as urea. One well-known genetic issue associated with the degradation of the purines is shown in that second red box, which is gout. And the gout is described in the text that comes with this image in the book shown. Here's a table of some other mutations associated with purine metabolism. The first one, adenosine deaminase, or ADA, was the first enzyme that was target of gene therapy in the early 1990s. The deficiency in this enzyme results in severe combined immune deficiency, or SCID. That defect is actually reflected in the function of lymphocytes. Here's a defect that I'm highlighting because Bill Nyhan, the co-discoverer of this syndrome, built a program at UCSD on inborn errors in metabolism. He became chairman of pediatrics in 1969 and built a program that is now known as biochemical genetics and is located in Hillcrest. This program has been a leading program in the area of inborn errors in metabolism. In this table are also two enzyme defects that directly affect the red cell. And once we have built the simulator we are about to, one could use it, for instance, to assess the consequences of incorrect function of these two enzymes. So I should say that we all have sequence variations in our genes, and we all have slight differences in our kinetic constants that characterize our enzyme functions. Most of these, of course, do not lead to any pathogenic states or serious difficulties, but some of them do, and that is often detrimental to the development of infants and often affects adult life if one survives to adulthood. So we're going to begin by forming a subnetwork for AMP metabolism. The set of reactions that characterize that are shown here. To the right of this module, we see the AMP exchange to glycolysis. To the left of AMP, we see all the processes that make and degrade AMP. I have a solid line on the outside of this box, which indicates a physical boundary with the surrounding plasma. The boundary on the right is dashed because that is going to go away when I integrate it with glycolysis. I've also separated this diagram by a horizontal dashed line that shows the degradative processes of AMP. Above it and below it, the processes that make AMP, including a salvage pathway, as we'll talk about shortly. We need to add new reactions to our model, and they are shown here, and I index them from 33 up, because that's what we had in because we had 32 reactions in the combined glycolytic and pentose pathway model, which we are now going to append with these reactions later in this lecture. Here are the concentrations of the intermediates in these pathways shown. These are known and have been measured, and I just note that these are very low numbers. These are low concentrations, and the fluxes through these pathways are very small. These pathways, like those shown here, that make cofactors, vitamins, and other low abundance metabolites tend to have low fluxes compared to the energy and redox generating pathways like glycolysis and the pentose pathway. For elemental balancing purposes, we can get out the elemental composition of these compounds just as we did before. AMP will flow out of this model into glycolysis, but as you see below the red box, there are other processes that will need to be coupled as we fuse the two models together. The stoichiometric matrix for the AMP network itself is shown here, where we will have internal reactions to it and then some of the exchange reactions. I note here that many of the exchanges of adenosine, adenine, inosine, hypoxanthine are measurable quantities, as these bases and nucleotides are being exchanged with the surrounding plasma. So by monitoring their concentration in the plasma, it is possible to get an experimental measurement of these. Here is the elemental balancing. As you see, all the internal reactions are balanced. The exchange reactions are not. The pathways through here are 5, and they are shown graphically on the following slide. I do know 5 fluxes here, as explained in the text, that allow me to calculate the steady-state flux map. And again, many of these are based on the measurements of the nucleotide exchange rates. Here I show these 5 pathways graphically. On top, we see 3 pathways that degrade AMP. One dephosphorylates first AMP. You take the phosphate bond off, you have adenosine, and that's secreted. The second pathway deaminates AMP to inosine monophosphate. That is then dephosphorylated to inosine, and inosine is secreted. And the third pathway is a different route to inosine from AMP, where AMP is first dephosphorylated, then deaminated, and then secreted. At the bottom, there are 2 pathways. Pathway 4 is just straight synthesis of AMP from adenosine that is imported from plasma. And you see that there is an ATP requirement for that reaction, because you have to add the phosphate bond group onto AMP. This will be coupled to the energy load in the integrated model in a moment. And finally, pathway 5 is a pathway where inosine is split into a base, and the pentose comes off, the base is secreted, the hypoxanthine. The ribose 1 phosphate that is generated becomes ribose 5-phosphate, which, as we'll see in a moment, is also an intermediate in the pentose pathway. That gets 2 high-energy phosphate bonds, and that allows us to drive the salvage pathway of adenine to AMP as shown. One can trace these pathways out by showing this little bubble here moving around. And this is often a useful representation when you have complicated pathway maps to look at. Okay, now we have the concentrations and the steady-state flux map. And as before, we can estimate these pseudo-elementary rate constants, or PERCs, and set this model up in a steady state. We'd like to simulate some of its responses before integrating it with glycolysis. And of course, our biggest interest is to see what happens when the AMP concentration changes. And given the simulations we have done so far, we are going to be interested here as an example to see what happens if all of a sudden the AMP concentration is increased as suggested by AMP efflux from glycolysis. If we simulate that, we can plot the results as follows. The first 3 horizontal graphs in this figure show the enzyme increasing through 3 fluxes in a row, and these happen to be all the fluxes that are in P2. So one would expect that in the new steady state that the pathway P2 would have a higher weight in recomputing the steady state flux map. And the three horizontal panels below show the concentration changes of the compounds affected by this change. So it would appear from the simulation that one of the degradation routes is activated. Okay, so now we have built a simulator of the purine metabolic network, or module, if you will, and now we need to put it together with the model we had before of glycolysis and the pentose pathway. That is shown here. Now I have a solid line all around my network, because now this is a complete model of the cellular metabolism I am going to consider. So this box here is what we started out with in the beginning, glycolysis alone. Then we put the pentose pathway on there, and now we will add the purine metabolic network. As I pointed out already, there are a few integration issues here. One is that ribose 5-phosphate is a common metabolite between these two pathways. The AMP node exists in both of these networks, so that will have to be fused into one. And that is simply done by adding up the corresponding rows in the stoichiometric matrix, combining them. This reaction here that formed PRPP was represented before by two ATPs becoming two ADPs, but actually what happens biochemically is both the high energy bonds on ATP go on to the sugar and AMP is generated, but that by the action of the adenylate kinase immediately equilibrates with ADP. That reaction is in the glycolytic model, and therefore I can have this detailed representation of this process now. As pointed out, as pointed out earlier, the synthesis from adenosine to AMP requires an ATP, so that will be a part of the energy load that a cell has. I should say a small part. About 75% of the ATP generated in red cell metabolism is used to pump sodium and potassium across the membrane and driving the sodium-potassium pump. But nevertheless, this pathway, even if it's low flux, represents a defined load on ATP. And then finally, we have all these exchanges with plasma that I mentioned earlier that are experimentally accessible, and we can measure them and get numbers for them. So these integration issues are also detailed in the text, and I think I've already gone through them. The AMP module needs, the AMP compound or the AMP node should become one. Same thing with the R5P node. Since it appears in two networks, it needs to be brought into one node. So all the fluxes that will go in and out of that node will need to be represented in that node map if you were to draw it. The detailing of the biochemistry here of PRPP synthase was just discussed. The load for driving AMP synthesis from adenosine was also discussed. This is a small load, but nevertheless now a defined load. And then there is are the exchange rates that we can experimentally measure that are shown here. Integrating two stoichiometric matrices is easy, although it gets bigger and bigger the more pathways you are considering. So this matrix looks a little unwieldy, but it is simply the systematic integration of the processes we've been looking at. So there we have glycolysis that we studied in chapter 11. There we have the pentose pathway that we studied first separately in chapter 11, and then we added. And now we are adding yet another block in here, which represents the purine metabolism. And you can see all the blocks below the diagonal that I formed by highlighting these two blocks has zeros in it, but those above do not, because there are compounds in more than one of these pathways that are common with them, and they participate in many reactions in both pathways. The null space now for this matrix is 8-dimensional. We had 4 dimensions in the null space for the integrated glycolytic pentose pathway model. And when we put the purine metabolism in here, we get 4 new dimensions in the null space. And these are, of course, ultimately related to all these exchanges of the bases in and out of the cell. Here we show these pathways on a network map. P1, P2, and P3 are all found in the integrated glycolytic pentose pathway model. Now, the AMP exchange rate, AMP input and output, that used to be just one pathway before, breaks down into 4 pathways. P4, 5, and 6, which are degradation pathways. They actually correspond directly to P1, 2, and 3 in the module model. Then I have the salvage pathway, which also appeared in that module. And now I have a futile cycle. A futile cycle are 2 reactions opposite to one another, where one of them generates a cofactor and the other one dissipates it. So in this case, taking adenosine to AMP requires ATP, but the degradation of AMP to adenosine simply is through dephosphorylation and splits off an inorganic phosphate. And therefore, if these 2 reactions are active simultaneously, it is basically hydrolysis of ATP to ADP and inorganic phosphate. That is known as a futile cycle. So there are the 8 dimensions in the null space of this stoichiometric matrix as I have chosen to represent them. We now carry out a simulation with this integrated model that is the response of the network to a 50% increase in the rate of use of ATP. And that is, again, bumping the PERK for that reaction by 50%. As shown in panel A here, the AMP node is now considerably more complicated. Before, we had just an input, a forcing function of a constant input, and then a turnover rate for AMP, a first-order turnover rate. Now, in the integrated model, we see that there are 4 reactions that can generate AMP and 2 that can degrade it. So in this case, I have chosen to show the dynamics of AMP as a phase plane of fluxes, where on the x-axis, I've summed up the 4 production fluxes, and on the y-axis, I am showing the consumption fluxes. So any point above the 45-degree line means net AMP consumption. If I'm on the 45-degree line, I'm in a steady state. If I'm below the 45-degree line, I am producing AMP. You see the reaction, the dynamic response to the perturbation given, which is a rapid rise in AMP consumption that we earlier interpreted as efflux. That turns around and drives towards a steady state line again at a different steady state, overshoots it, and then you get another overshoot, and then it settles down at that point. So this will have a damped oscillation type appearance on a time response plot. Panel B is a slight modification of panel A, where I simply sum up all the fluxes going in and out of the node to get a net flux in and out of the node. So at steady state, that net has to be 0. And now if I'm above the horizontal line formed by the line going through 0, I will have consumption. If I'm below, I will have production. And so that same response as shown in panel A is shown in panel B, and you just have a slightly different view of it. With Mathematica, it's relatively easy, and I presume with other packages as well, to obtain the same response from the model combining glycolysis and the Pantos pathway, and comparing that response to this model that includes the AMP module. So we can basically plot the response plus minus the AMP module. And this is what you get. You see the dynamic responses. The full model here is the solid line, and the dashed line is the simpler model of glycolysis and the Pantos pathway. And you can see that the ability of AMP to be degraded or synthesized, otherwise bounce back and forth in and out of that module that generates or degrades AMP, dampens the response. The response is harder, if you will, more stringent, more strong, if the forcing function is a constant. Here it's no longer a constant as it was before, but is actually determined by that entire subnetwork. And you see a much more dampened response from the full model. Now you have a model of 3 pathways working together, and you can now perform all sorts of simulations to study the physiology of this cell. This lecture simply shows the model building process and shows this simulation as an example of what you can do. At the end, I will comment on the fact that we are well on the way to building a meaningful whole cell model of the metabolism in the red blood cell. We of course know that the red cell circulates in the blood, picks up oxygen in the lungs, and delivers it in the tissue. This cell is enucleated. It doesn't have a mitochondria. It's just a single compartment. So it's not compartmentalized. And over 90% of the protein content of the cell is hemoglobin. And that is easily visualized by the image I had from Goodsell's book in an earlier lecture. So this is a cell that cannot grow. It circulates through blood and picks up oxygen and delivers it in the tissues. It is also involved in trafficking of CO2 and has a few other functions in the body. But its metabolism is very simple. And now we can begin to view the model we have built as a model of a metabolic machine, in quotes, that meets metabolic demands, shown to the far right. In this cell, glycolysis, nucleotide metabolism, and the Pantos pathway are the major active metabolic pathways in the cell. There are others, but these represent a fair amount of the metabolic activity in the red blood cell. And we also have that unique bypass on glycolysis that makes 2,3-DPG, as we will discuss in some detail in the next lecture. So you see on the far right, we have the loads on the cell, or the demands. I mentioned the transmembrane pumps that need to be driven, mostly the sodium-potassium pump. Nucleotide metabolism is involved in maintenance and repair, maintaining the AMP level. And this function is, for instance, very important when it comes to blood storage, when the adenosine nucleotides tend to be lost over time. We have NADPH, or glutathione, dealing with oxidants. An adult male of 70, 80 kilos or so may have 5 liters of blood circulating, and at a 40% hematocrit, that means that there are 2 liters of blood in an adult male. So that would be about 2 kilograms, a little over 4 pounds. And women, of course, correspondingly less. But this is a large volume of cells, and it has high glutathione concentration of 3.2 millimolar, as we saw in the chapter on the pentose pathway. And this represents a massive reserve of redox potential that is circulating in your blood. And any oxidative challenge to the body is often met by the red cell and would lead to drainage of the charged state of those cofactors. The iron in hemoglobin is active in the +2 state and is constantly being oxidized to the +3 state, where it is non-functional and leads to what's called methemoglobin that cannot bind to oxygen. NADH is used to constantly reduce iron to its functioning state. Finally, as we'll talk about in some detail in the next lecture, 2,3-DPG is a ligand to hemoglobin that regulates its affinity for oxygen, as shown in that little plot that's inserted in this diagram. So here we have a metabolic machine comprised of a few pathways, and we have multiple demands put on that metabolic machine as indicated. These all have to be simultaneously met. They can all be disturbed separately. They can all be increased or decreased separately in various physiological states, and the red cell has to react to those. Sometimes it cannot react, as I indicated earlier. For instance, glucose 6-phosphate dehydrogenase deficiency, a very common deficiency in the human population, leads to impaired ability to drive flux down the pentose pathway, and therefore an impaired ability to deal with loads on NADPH. We now see why we are interested in all these pools and ratios that have been introduced throughout the lectures, because those pools and ratios would give us a physiological interpretation of the function and state of this metabolic machine. Much more of a bioengineering view than a biochemist's view. Finally, I just thought I should say a few words about how one can study genetic defects using a model like this. The process goes as follows. First, a sequence variation is detected, a SNP as it's called, single nucleotide polymorphism. And here's an example of one that may hit hexokinase, and that little gray insert shows a lower Vmax for the mutated form, or for that I should say variant, sequence variant. The function of that enzyme is now placed in the context of a network model, and we can see how a reduced Vmax or altered kinetic properties of one link in this network will change its properties. And we would have to calculate a different set of perks, clearly, for this situation, because that defect may lead to a slightly different steady state. Some of these defects don't lead to a notable difference in the steady state under normal physiological conditions, and you'll only see them when you are perturbing the network. Some defects, like those mentioned at the beginning of this class, will have the red cell in a different steady state under normal conditions. But in any event, if we have a genetic variant accounted for as shown, we would have two models, one with it and one without it. And then we can challenge these models and try to understand the differences in the physiological response. In this case, a SNP and hexokinase will impair the ability to make ATP, and that, for instance, would impair the ability of the cell to respond to an osmotic shock, and a shock which a normal cell might withstand but the cell based on this genetic variant might not be able to respond and would actually swell and eventually lyse. And that would be then a pathological state resulting from that altered genetic property. Here is one specific example of such a mutation indicated at the bottom. So we have a particular mutation there in position 1667 from T to a C, and that changes the kinetic properties of hexokinase, and that leads to reduced glycolytic flux and ability to make ATP, and cells that have that variation in the hexokinase have an impaired ability to deal with osmotic stress. So this shows how these dynamic simulators we've been talking about can be used not only to look at changes in environmental parameters, but also genetic parameters. And of course, studying them at the same time is of great interest. So, a few summary points here at the end. Purine metabolism is somewhat complicated as it involves a lot of degraded pathways and biosynthetic pathways, and it has many genetically based pathological states associated with it. The nucleotides are synthesized and degraded to be in a steady state that is dynamic, constantly making and degrading AMP. And I showed this with a node map with 4 reactions going in and 2 going out. And the results of this is an improved ability to respond to dynamic perturbation rather than the more rigid situation which we considered earlier, where the input flux of AMP, the forcing function, was simply a constant. We can build a subnetwork for AMP metabolism and look at its responses. We can then integrate it with the model built in chapter 11 that describes glycolysis at the pentose pathway. There are many points of contact between these, as explained, and we have to deal with these integration issues as we build the integrated representation. The number of pathways that characterize the null space grows. There are 8 in this case, but we do have experimental measurements on inputs and outputs that allow us to fix the length of each one of these pathway vectors in the steady state. So summing them all up leads us to the steady state flux map. And then the model can be simulated and the results are studied. And finally, as I spoke to at the end, this network model is not large, but represents important aspects of red cell metabolism. And we can continue to build more modules into this model to make it more and more realistic as we expand it in scope. The following 2 lectures will show how that is done for proteins in this network. We will build a module for hemoglobin itself and for one of the regulatory enzymes, key regulatory enzymes in glycolysis, the phosphofructokinase, and see how those can also be fused or integrated with the network model. One can thus continue to build such modules that one has information about to expand the size, the applicability, and predictive power of a whole cell simulator. Thank you for your attention.